Hello, everybody. It is great that I have this opportunity. I especially thank GenScript for supporting this webinar. Today, I'm going to talk about advanced genetics, which is about mosaic analysis tool and short conditional introns. This work has been led by two brilliant scientists, Mingyu Young and Sam Wu, who's been working in my lab over the last 4 or 5 years. So, before we start, I'd like to start with some philosophical questions. So, is science fun? And I think all of us are working in the field of science because we love it. However, we are also forced to think about cool hypotheses, new ideas, and we are also asked to work really hard. And at the same time, I remember that when I was a young graduate school student, I was feeling with the many hours work, feeling very tired and also feeling hungry almost all the time. So when I was working so hard, I was always feeling that whether this is really fun or not. And I think it is actually coming from the way we do science. And the reason that I'm talking about is if you think about the way to do science, I think there are three different ways. The first one is hypothesis-driven research, and I think this is the most popular way to do research, and especially our funders are asking us to create new hypotheses and then propose some work to be carried out with the research budget. But I feel like this is a way to look for an expected result because hypothesis is actually coming from your brain, which means that it is based on the fact that you already know. So it is usually not so easy to find some new discoveries with hypothesis-driven research, which is my opinion. And secondly, scientists also love to work with the curiosity-driven research. And this is probably the mostly well appreciated research type by many scientists because we can then work on something that we are interested in. And the last one that I can think about is technology-driven research. And I now believe that this is a type of research that we have to pursue, especially when we are looking at the unknown territory. And so far, I've been using these three different type of research uh, method. I've been using these three different type of research methodology, but I think now the one that is fitting very well to myself is probably the technology-driven research. And if I talk about this in this way, people may think that technology-driven research is something which became possible after we get into the 21st century. But the truth is, the technology-driven research was one of the main way to drive the science into the future. So, for example, Galileo, a famous astrophysiologist, has made telescope first before he made an observation on the small stars that are moving around on of Jupiter. And later, after making this observation, he then realized that these are the four moons that are turning around Jupiter. And for this fundamental discovery, his technology, telescope, was very, very instrumental. And lately, we also know from the examples of LMP and the founding director, Max Perutz. As you may know, that LMB is an institute at the University of LMB is an institute at Cambridge in the UK. The LMB has been nicknamed as a Nobel Prize factory because 12 Nobel Prize has been won and 11 other Nobel Prizes were given to its alumni. And Max Perutz once said that discovery cannot be planned. They pop up like puck in unexpected corners, meaning that he's somehow trying to defy people who are advocating hypothesis-driven research. And LMB has been making technological evolution by introducing DNA sequencing, protein antibody engineering, X-ray crystallography, confocal microscope, and cryo-EMs, which has also been given the Nobel Prize. So I'm not working at the R&D, but I think it is a very nice example that they are doing technology-driven research so that they can also advance the way we do science. So I am a geneticist, and I'm very interested in developing new genetic tools so that we can do advanced genetics in the lab. Um, Conventionally, the genetic model organisms has been yeast, fly, and mouse. And in the 20th century, we have observed revolutions in molecular biology and biotechnology. And in a short summary, it has been really crucial to start using E. coli for plasmid construction. And this has been also very, very important for all kinds of protein production. And viruses have been the teacher of us for various molecular biology tools. And we also have performed for the genetics using C. elegans and zebrafish. And in the late 20th century and in the beginning of 21st century now, we have now observed many exciting new advancements in the stem cell biology. For example, using mouse embryonic stem cells, we were able to create knockout mice. Also using human pluripotent stem cells and iPS cells, human stem cell biology became possible. And more lately, we have also observed that people start working with organoid to mimic the in vivo physiology in an in vitro system. And at the same time, you'd all know that genome engineering tools have been rapidly evolved so that before people were using meganucleases, zinc nucleases, and talents. But now, as you all know, CRISPR-Cas technology has revolutionized this field of research. And I think that the two technology, stem cell biology and genome engineering, is the two main technologies that are currently driving our bioscience into the future. So I just talked about intestinal organoid, and the reason that I started working with the intestinal organoid was this could be used as a very nice model system. We know that people have been working with invertebrate models, vertebrate models, and also with cell lines. But I can also tell you that organoid could also be a nice model system because here we can observe in vivo phenotype in an ex vivo system. They are ever-expanding system as if they are cell lines, so you can start culture from mouse tissue or from human tissue. You can also cryopreserve them whenever you would like to do so. And it is also easy to treat them with ligands and inhibitors. They are evolutionarily very close, so you can work with the mouse, or you can even do a human experiment in a dish. However, about 12 years ago, when I observed organoid for the first time in Hans Clever's lab in the Netherlands, it was not easy to do any genetic engineering in organoid because we even didn't know how to transfect those genetic tools into the organoid system. So in the last 10 years, I've been working on organoid to advance its genetic system. So in summary, I have firstly made viral gene overexpression and knockdown method in organoid. We also have introduced how we can generate bacterial artificial chromosome transgenic organoid. And this was very crucial work with which we realized how to do a proper transfection in the organoid system. And later, when we got to know CRISPR-Cas technology, we were able to apply the technology immediately to organoid system because we knew how to do organoid transfection. And this work has led us to do gene correction in patient organoid for the first time. And there was a cell stem cell paper in 2013. More lately, although this is not my work, but one of the founding member of organoid field, Toshiro Sato, his lab reported that NEPA21 electroporator can be very useful for highly efficient transfection in organoid system. And using this technology, my lab was able to show that piggyback transposon system can be very useful for controlled gene expression in the organoid. We also have introduced how we can generate conditional knockout in the organoid system. And more lately, we also have worked on CRISPR-Cas technology screening. Uh, However, some other labs also already published some work reporting that CRISPR-Cas screening is possible in intestinal organoid and also in the other organoid systems. So up to now, what I can tell is that the organoid system and the related genetic engineering tools and some transfection method have been fully established so that one can think about using organoid as a model system in the lab. So I feel now that my contribution on this part is nearly over, because now we can easily work with organoid for every purpose that we like to in the lab. So with all the new technologies that I developed for the organoid system, I started thinking about how we can work better with the mouse system, because initially I was trained as a mouse geneticist, and it was always a little bit mixed feeling to see that mouse model is becoming less attractive compared to the fascinating organoid system. So with that in mind, I started thinking about what are the next for human disease modeling, and then I start thinking about some of the limitations that we have with the current mouse genetics. So these are the three thoughts that I had. So first one is, in the human, disease do not occur in a systemic manner. However, when we do mouse modeling, we are knocking out the gene in the entire body or in the entire tissue. So this means that to study some features in some features of human disease, such as cancer and aging, that do not happen in a entire tissue, we may need to start working with the mosaic genetics tools. And secondly, you all know that mice are not human, so that is the reason that we are working with the human pluripotent stem cells and organoid. But there are also people who are thinking about using primate model system. However, as you all know, for example, conditional knockout method is available in the mouse, but doing the same in the other species is still very challenging, and I think that it is not yet available in the primate model system. So short conditional intron method has been developed in my lab, and this is one of the topics that I'm going to talk about today. And as a last one, this is another thought that I have. However, we do not have an answer yet for this question. The thing is, when we do genetics, we are not thinking about the reversibility and irreversibility. For example, many drug candidates are focusing on the cause of the disease, but we do not know yet whether removing the cause can actually revert the outcome of the disease. For example, when somebody has a dementia, if the person has lost all the neurons, then uh, removing the cause that has caused dementia can be reverted. I don't think so. So I started thinking that the current genetic method that we are using in the mouse doesn't allow us to test what if we restore the gene function after performing knockout. So I think that we need to have a reversible genetics tools so that we can also test the reversibility of the phenotype, and when is still the window time that we can cure certain disease. So these are the three thoughts that I have, and today I'm going to touch on the number one and number two. So today's main topic is about mosaic overexpression system, and the short conditional intron. And due to the limited time, we also developed a mosaic knockout system, but I'm not going to talk about this point. So, as you know, cancer develop in a sequential manner, and we all know that cancer starts from a single cell that becomes a mass. However, when we talk about cancer, by definition, we should be able to see a mass, and there should be some features of dysplasia, because before this point, I think medical doctors will not call them as a cancer. And after that, they as a cancer. And after that, they start showing invasive phenotype and sometimes metastasis. And obviously, current focus of cancer biology is on this part, whereas we have very little knowledge on the precancerous transformation because it is very, very difficult to observe what's going on in this part of the cancer development. So the reason is that in the clinic, the initially mutated cells are histologically very similar to the surrounding tissue. So it's nearly impossible to study and find them to study their behavior. Secondly, when we model this in the mouse, we've been knocking out tumor suppressors and activating oncogenes in the entire tissue so that we have not really utilizing mosaic method in order to see the behavior of the mutant cells. So to overcome this two weak points, we have to work on fluorescence labeling of the mutant clone. And I think that we also have to have a tools with which we can precisely make a genetic modification in a single cell level. And moreover, thanks to the single cell RNA-seq technology, if we have a small lesion of a mutant, we can isolate this part. And then after a little dissociation, one can profile all the RNA expression pattern of the mutant and surrounding wild type as well as the mesenchyme and immune cells in the environment all together using single cell RNA-seq. So if we can perform single cell RNA sequencing, then we can also detect some of the changes that are only happening in the very beginning of the cancer development. So with that thought, we started thinking about some technological advancement with which we can cover these three requirements. And I came up with an idea to modify ROSA26 confetti allele, which was initially developed by Hans Klever's lab. And here we have a stopper which blocks the expression of the lineage tracing cassette that contains 4 different fluorescence colors. And what we have done is we modify this RP in a way that it can accommodate another cDNA in the downstream of it. And by doing so, we now have a red to cDNA system where the cDNA is co-expressed together with RP, meaning that if the cDNA is an oncogene or an additional recombinase or transcription factor, we can only express those in a red clone specific manner. So using this method, we can achieve fluorescence labeling, red clone specific oncogene expression, and this system is compatible with the single cell RNA-seq. So we generated 3 different mouse lines, namely R2-Notch1, R2-KRAS, and R2-PI3 kinase. And this is the example of a red-to-KRAS system. So on day 2, after tamoxifen administration, we can nicely see salt and pepper pattern of 3 different colors, meaning that all 3 clones are induced in a very equal manner. However, if you just wait about 2 weeks, you can clearly see this red clone domination due to the fact that they are expressing KRAS oncogene, and thanks to the activation of proliferation, they can occupy the tissue with its own progeny that are containing the red clone as well as the KRAS expression. Interestingly, the tissue architecture is still looking pretty normal, but you can appreciate how rapidly the tissue is turning into the red. We confirmed the system by looking at the downstream activation, such as Notch intracellular domain, phosphor-ERK, phosphor-AKT. And another nice point is that, like the original confetti allele, all these modified alleles are behaving in a very similar way in terms of yellow, red, and cyan clone generation in the tissue upon pre-activation. So we firstly look at the yellow clone and the mutant clone behavior in a separate way. So in the intestine, we have a crypt and villus, and under the crypt, at the base of the crypt, we have stem cells that are competing each other for the space of the niche. And over time, if we have a label here, then one stem cell can increase their clone size into 2 or more, so until it occupies the entire crypt base. And this has been nicely observed in the confetti system, so we already know how they look on day 4 to 1 week to 3 weeks, and this is the clonal size distribution in the tissue. So if we look at the yellow clones, which are wild type, they always follow the same pattern of confetti. Whereas if we are looking at mutant clones from these various mice, we see clonal expansion more rapidly from the red clone specific manner, meaning that if we have a Notch or KRAS PI3 kinase activation, this will drive the clone to grow faster and more aggressively so that they can occupy the entire crypt with its own progeny in a faster way. However, this was already studied in the past with a different method. So up to now, I would say that we didn't really find anything novel, even after having made this mosaic analysis tool. However, my postdoc, Bon-Kyoung, has found something very interesting because he was looking at the red clone that is red and yellow clones that are next to each other. So if we are looking at the yellow clone that are far away from the red, they of course behave very much like the original confetti allele because they just have a wild type behavior. However, when the yellow clone was sitting next to the red, we found that their clonal distribution is somehow strange, and they were looking to be going faster to the monoclonality. And this means that something is, secreted from the red, with which we can they can change the behavior of the yellow clone, So we were very much interested in this biological phenomenon because this was a very unexpected observation after we made this mosaic genetics tool. And my collaborator, Professor Ben Simons, is a biophysicist who can understand how the clones are competing each other in the crypt. And based on his theory, what we could learn is that the time that is taking for the clonal competition is correlating to the number of stem cells and also the speed of the stem cell competition. So we can basically check the speed of the competition by looking at the cell proliferation, and we can also count the number of stem cells to see which of the two factors are mainly affecting in the context of the R2 onco models. So when we check the yellow clones with the proliferation markers, we found that the yellow clones that are away from the wild type, the mutant, or close to the mutant, have a very similar rate of proliferation. So there was no difference in this case. Whereas when we check the stem cell number, we found that the yellow clones have a much less number of stem cells compared to is the is the wild type clones that are far away from the mutants. So this means that somehow the red clone here is secreting something with which they can reduce the stem cell number that is sitting next to it. And to know what are the factors that are influencing the wild type clones that is sitting next to the mutant clone, we performed single cell RNA sequencing to profile not only the red and yellow epithelial cells, but also all the mesenchymal and immune cells that are surrounding the tissue. And we have done this for the red to onco models, as well as the confetti models as a control. We have done a lot of single cell RNA-seq analysis, but just to tell you the conclusion, I'm showing this slide that where we can observe epithelial cells, mesenchymal cells, and immune cells. And after analyzing all these cell types, what we have found is that in the epithelial cells, two BMP ligands, BMP2 and BMP7, are highly unregulated in the KRAS and PI3 kinase active mouse. And interestingly, in one of the stromal cell population, which we named as a STC2, showed a very nice regulation of Wnt agonist, soluble FRP2, and R-spondin 3 was somehow reduced in this population. And this is happening in a very specific manner in the PI3 kinase model only, which means that on top of the BMP elevation in the PI3 kinase model, the red clone is also influencing the STC2 cells so that they cannot support Wnt activity to the microenvironment. So we went on some biological validation to see what we have observed is true. So in the KRAS model, we found that there is a high amount of BMP secreted by red clone. In the PI3 kinase model, we saw the same BMP, but at the same time, we found the mesenchymal population to be compromised in supporting the Wnt activity for the yellow stem cells. And to model this, we take out Nogin to mimic the BMP high condition, and Nogin and R-spondin to mimic the BMP high and Wnt low condition. And as you see in both conditions, the organoids cannot grow nicely well because they are losing their stem cells, as we have observed from the yellow clones that are sitting next to the red. However, when we have introduced the mesenchymal population of the wild type, we saw that this mesenchymal population can rescue the phenotype. But interestingly, when we isolate the same mesenchymal population from this PI3 kinase model, we found this mesenchymal population do not have the niche supporting activity anymore, because we also cannot see the increasing number of organoids compared to the wild type case, meaning that the mesenchymal cells, especially the STC2 in the PI3 kinase model, are indeed compromised in providing proper Wnt support. And secondly, we also wanted to check out the BMP to see whether this is really the ligands that are mediating the crosstalk between red and yellow in vivo. So for this experiment, we have used a well-known chemical inhibitor, LDN, to block the BMP receptor activation. And we basically did same lineage tracing and treated LDN to the mouse to see how the lineage tracing pattern is changed. As I mentioned earlier, this is the clonal distribution of confetti mouse. And when we look at the yellow clones that are next to the red, in the KRAS mouse and PI3 kinase mouse, the clonal distribution was more to the monoclonality. However, after treating the LDN, this was coming back to normal once again, meaning that if we block BMP, we can alleviate this phenomenon. When we check the statistics in a different way, we saw the same, so that with a In this mouse, there is an elevation, but with LDN treatment, we can suppress this back to normal once again. So this confirms that BMP is the ligand that are mediating the crosstalk between red and yellow. So after generating this mosaic overexpression model, we were able to show that the red clone in the tissue, once they have an oncogene activation such as KRAS and PI3 kinase, they can suppress the stem cell activity of the surrounding tissue. And we also found, especially in the PI3 kinase case, the red clone is not only secreting BMP, but they also influence the mesenchymal population so that they cannot support Wnt activity anymore for the yellow stem cells. So I hope that I could show one very nice example how we can use a mosaic overexpression system to really visualize the very first event of cancer development in the mouse. Now I'm changing my gears to short conditional intron system to show you how we can generate conditional knockouts in a different way. Even after CRISPR revolution, people now say that the conditional knockout is still not really solved because initially, right after we have a first wave of CRISPR-Cas technology, Wang et al. Reported in Cell that we can insert two LOXP sites with a simple CRISPR by introducing two guides in each side of an exon. He said that we can introduce this LOXP site very easily. However, more recently, Gru and his friends mentioned, reported in Genome Biology that actually this strategy works in a very inefficient way, so that they were only successful with less than 1% of cases. So this means that someone has to work on this problem so that we can have a conditional knockout in a more efficient way. So we think that we are one of the people who are actually solving this problem. What we have made is a short conditional intron, which we call as a SCON. And using this method, the method is using a conditional intron that is only about 189 base pair long. So if they are in a EGFP cDNA, they have a perfect splicing function so that this intron can be spliced out, and then you can still observe very nice protein expression. However, after Cre recombination, the intron can lose one of the important functional groups, such as branching point, and then the splicing function is compromised, and then you don't see the protein function anymore. So we checked it in 293 cells that normal SCON nicely allow the GP expression, but after it is recombined, there is no expression of GP. And when we compare this green fluorescence level, we found that the SCON containing EGFP has a very identical FACS plot, FACS intensity compared to the EGFP case, meaning that when the SCON intron is inserted, it doesn't really compromise the gene expression. However, after the Cre activation, the brightness is completely shut off so that you can compromise the gene expression perfectly well. So with that success, we moved on to mouse genetics, mouse generation, to knock out beta-catenin, which is called Ctnnbi1, using the SCON method. So we basically made SCON intron flanked by a very short homologous arm, which is just about 50 base pair each. So overall, now the oligo is just about below 300 base pair because it contains 50 base pair of each arm and 189 base pair of intron. And with that, we were able to make a nice insertion using CRISPR-Cas technology. We checked their recombination site to see that all the sequences are correct. And we found that a modified allele could exist as a homozygote, which means that since we know that beta-catenin is a lethal gene, having the homozygote in a healthy manner, meaning that we have achieved conditional knockout very nicely well. So we moved on to functional validation. So as I mentioned, the expression level of beta-catenin is very comparable between homozygote and heterozygote. And after tamoxifen administration, you can nicely see that the homozygote has lost the beta-catenin expression completely. And this has lost leads to the depletion of proliferating stem cells and progenitors, so that on day 5, the entire epithelia was gone. So this was a very nice confirmation that our SCON technology can be used for generating a conditional knockout in the mouse. And we have now moved on to other species to see whether similar conditional knockout strategy can be applied. So here we have used various cell lines from various species such as rat, 2 species of monkey, and sus is a wild pig. And as you see that the both EGP and SCON contain EGP, they show very nice GP expression pattern. And after Cree activation, you can nicely see the knockout of the splicing function. And we also checked that SCON intron has a functionality also in xenopus as well as in zebrafish, meaning that the same intron, conditional intron, can now be used in generating conditional knockouts, probably in many mammalian species, but also in other species living in the water. So with that, I now come to my conclusion. So I had 3 thoughts that I explained in the beginning, and I can tell you that the mosaic issue was already solved by our lab by generating these rat-to-onco models, but I can also tell you that we are currently preparing a manuscript for rat-to-knockout, with which we can do a mosaic knockout in any tissue that we want to. And secondly, with the SCON method, now we can generate conditional knockout mouse much more rapidly using CRISPR-Cas technology. However, this same technology can also be used for various other species. So it may solve an important issue we have now. And thanks to the SCON technology, we can now think about some other conditional intron of which activity can be activated from inactivation form, or even as a reversible artificial intron. So we are currently working on this point to see whether we can generate some other conditional introns that can be very useful for the genetics field. So these are the research area of our lab. We are biologists working on intestinal and stomach stem cells, and also interested in Wnt and E3 ubiquitin ligases. However, at the same time, we are very fond with genetic tool development, such as mosaic analysis tools, short conditional introns, and we are also working very hard with the organoid technology and so on. So I hope that if you are interested in one of these themes of research, I hope that I can be contacted by you in the future. And this is my lab members, and I would like to say my special thanks to Dr. Seung-min Han and Min-Kyu Young for the first part of the work, and my PhD student Sam for the second part of my work, and also my collaborators and all the funders as well as the IMBA who've been very nice support, which has been very supportive for my research team. Thank you for your attention.
Thank you very much, Dr. Koo. It is now time for the question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. Okay, Dr. Koo. So to get us started for today, I'd like to ask you, how relevant is the presented mouse study in the context of human cancer development?
Yeah, so it's a good question, but it's always very difficult to say that we can model human cancer perfectly well in the mouse system. So if I say yes, we can do, that's probably a lie. But at the same time, we know that we cannot model everything in human setup because basically we cannot use human for experimentation. So we only have a mouse as an option for now, to be honest. And there are some organoid models that we can still utilize to see how cells are interacting with each other and so on, but they do exist in a very artificial format. So we have to understand that they are, on one hand very useful, but on the other hand, it also comes with many shortcomings. So at the end, we have to understand what is possible with the mouse models. Somebody has said that all models are wrong. So in a sense that the organoid models and mice models, they can never be the human. So in my opinion, I think I have already explained well during my presentation that mouse model is probably a nice proxy when it comes to the cellular interaction within a tissue in a very intact manner. So that is why we were trying to develop this mosaic analysis tool so that we can observe those behaviors of mutants in real setup for the first time.
The next question asks, why do we need a mosaic system? Can this strategy be used for any other disease modeling?
Yeah, so I do believe that the mosaic system can be used in many different disease models. To be honest, even I myself was wondering what could be done with the mosaic system when we started working with this tool. This is pretty much like, the question that Galileo had. So he was building this telescope, but you can also read about his thought that he was not sure where it can be used. So actually, what I can tell you is that the more you start using this tool, the better understanding you have on where to use it. So far, the genetics has been very simple. I think people were knocking out genes in a very easy manner in the entire body of the mouse or the entire tissue. But we have to admit that this is not really happening in human. I mean, we do have some germline human mutants, but most of the somatic mutations are happening in a mosaic manner. And there we have to think about the nature of cell-to-cell competition, because all the wild-type cells are naturally competing. And if we have mutant cells that are aging, or they are going for becoming a cancer, then the behavior of their interaction will be different. But without having worked on mosaic models, there was no way for us to understand this. So I do believe that it's now time to think about the maximum the capability of the tools. It is now available. I can also tell you that we have also achieved a mosaic knockout system in the lab, which will be coming along pretty much soon, I hope next year. So once we have that, then I think people will start reading the mosaic genetics that have been done in Drosophila, which was also very popular at the time. So I hope that I am one of the first persons who are opening the doors for the mouse mosaic genetics, and I wish that I can be contacted by many of you so that you can also try the mosaic system that we have developed together.
Thank you very much. And now we're getting some feedback from your comments as well. There's a follow-up question here, which perhaps is interesting for you to clarify. And it asks, why do we not have use a more relevant in vitro model, since mouse models have their limitations in mimicking a human one? I mean, you kind of addressed that already, but would you have any further thoughts?
Oh yeah, so I can tell you one major difference between the organoid system and the crypt that are residing in the in vivo situation.
So if you grow organoids, these stem cells, they are not at all in the limited space anymore because they can expand all the time. So in vitro, there's no space restriction. Whereas in vivo, the intestine is completely filled with crypts, so that they are really competing each other. And that is why we can observe crypt fusion and fission events. Whereas in organoid system, we only see crypt fission. They do not fuse together unless you intentionally mix them in a very small, tiny wells. So thereby, there is a difference. And I do think that we are basically underestimate the importance of cellular competition in the tissue. And this is actively ongoing. You know, the hematopoietic stem cells are also competing each other to take more part of the hematopoiesis. This is basically happening in, I think, in almost all adult stem cells. And which one gets which mutations and how they survive after acquiring that mutations, these are totally unknown. And I think it also has a very nice connection to oncogenesis as well as the aging process, because we also do not know, which cells are, undergoing faster aging and undergoing faster transformation compared to its peers in the tissue. These questions cannot be addressed using organoid system where the space is not limited. I think somebody may make the organoid system a little bit better by planting that in a micro device, fluidic device, where the space is limited. Then maybe we can observe something similar that we can observe in vivo. Until that time, I think mouse system still has some advantages over the organoid systems.
Thank you very much for that clarification, a point worth making, I think. The next question asks whether this strategy can be used to target, for example, targeting non-epithelial cells in the intestine.
So the question is about targeting non-epithelial cells, right?
Correct.
Yeah, I think that part is not been done, and at the same time, it is also not really feasible at the moment because we are lacking useful clear lines with which we can target the non-epithelial cells in the intestine. So this is something I think we have to really think about from now on to utilize the model in different tissue and different cell types. But I can tell you that the system itself, the genetic model itself, is universal, so that whichever cell types or tissue that you like to apply to can be used. So this doesn't limit the questions and ideas that you want to have.
Okay, thank you very much. A quick reminder to our live audience, you do still have time to ask a question. To do so, please type it in where it says type your questions here, and then press submit. So our next question, Dr. Koo, well, she actually asks if you could further clarify and explain differences in global expression and in tumor-induced expression of inhibitors from tumors in stem cells to normal yellow stem cells.
The global gene expression change is the question. I just didn't get the question.
The question is the difference in global expression and tumor-induced expression of inhibitors from tumors from stem cells and the normal yellow cells.
It's not really clear to me, but I think I got the question occasionally. So we've done single-cell RNA-seq analysis to see the changes between yellow cells and red cells, but the analysis is very complicated one because, as you know, the yellow cells, as the normal cells, they have from stem cells to all kinds of differentiated cells of the epithelium, and the same is true for the mutants. So the red cells also contains stem cells to all the other differentiated cells. So then you have to really put them together. And then after that, you have to look at individual cells to see whether there is any gene expression changes individually. So if you have, let's say, 10 different populations, then you have to get 10 different differentially expressed gene lists. And then the BI and myself and my postdocs had to go through all the gene lists to see, what are the changes and so on. So it was not so easy to pinpoint the causative genes, but we somehow managed to come up with an idea. So at the end, the single-cell RNA-seq analysis part was very shortened due to the time limit. But I can tell you certainly that there are, lots of information you can dig into. But we were at the same time very lucky to find out some of the effective ligands and inhibitors that we found from the list. Yeah. So the answer is it's very complicated.
It is indeed. Our next question asks if we can use this technology to study metastatic niche.
Yeah, I think so. But that means that we are applying this technology after the tumor onset. So this will again make the genetics again very complicated. So I myself was not thinking to do so because at the end it's too complicated because the confetti system is already very complicated. And if you want to do that, then just for the mouse matings, you may need to spend half of your PhD time. So that is why we didn't go that part, but there is certainly an interesting things to be done. So yeah, I think that is now up to the people that what kind of things you want to address with this system is really up to other researchers.
Okay, thank you very much. Our next question asks, is this out-competition strategy occurring only in an oncogenic system, or does it apply also within a wild-type stem cell?
Yeah, so if I have understood the question correctly, my answer is yes, that it cannot be applied to other systems. So the example I showed today was the transformation being started with one single oncogene expression. But the same oncogene can be used to see the effect of a certain signaling activation in developmental context. Then I would say it is not oncology anymore. It is more for developmental biology study. And I also mentioned briefly that we have also managed to make a knockout system. So once that becomes available, I believe that this mosaic system will be expanded to many different questions in biology. So for now, we only made oncogene series. Sorry for that. But more exciting tools are coming along. So please stay tuned.
Many thanks for that. And then finally, I'd like to ask you, how does the presented study influence future cancer prevention strategies, do you think?
Yeah, this is something very exciting. So before we were looking at the initiation of the transformation. I mean, this cannot be said as a transformation even because transformation is when we have a cancer. So this is pre-transformation, let's say. So in this pre-cancerous transformation, not much is known because so far we could not study this well. And not only our paper, but also two other papers were published together in Nature in the middle of this year, and the messages were pretty similar to each other. So I was actually very surprised, pleasantly. And the message is that these cells are competing each other already with the normal cells, even before they show transformed characteristics. this means that there might be an intervention strategy before we even see a cancer mouse. It means that maybe at some point, like we are taking some pills to kill parasites in the intestine, we might be, taking some pills to kill transforming cancer seed in the near future. Who knows? But I certainly think that this is a very exciting opportunity. So at the moment, I can tell you that our lab is currently focusing on how we can utilize some of the cancer drugs that are already existing in order to apply those to our modern system to see whether these drugs are already effective in blocking the mutant behavior. Which, if that comes out to be true, it will be pretty exciting, I think.
Thank you very much, Dr. Koo. That was an exciting note to end on. And that is where we're going to need to finish for today. I would like to thank you very much, Dr. Bon-Kyoung for your presentation and for answering questions today. I would also like to thank the webcast sponsor, GenScript Biotech, and of course you, the audience, for taking the time to be with us today. You will shortly be redirected to a short 4-question survey, and we would appreciate it if you could give us your feedback. Remember, you can watch this webcast again at any time on demand at nature.com/webcasts. Thanks for watching, and I hope you'll join us again soon.